Good afternoon. My name is Haylie Easley. I'm a junior pre-med major, and today I'll be presenting on the pharmacological target identification via cell transduction analysis of the minimal phenotype cluster in autism spectrum disorder. I would like to thank the judges for the opportunity to present my work, as well as for the use of the podium. There are no conflicts of interest to disclose for this research. This work builds upon the efforts of Daniela, Taryn, and James. The clinical problem. Current treatments for autism spectrum disorder, also known as ASD, utilize a one-size-fits-all approach, managing ASD as one condition. This type of treatment is ineffective. ASD is not one condition, it is many. Different genetic variables present as many different symptoms that differ per each person. Current data shows 1 in 36 children are diagnosed per year, with an increased prevalence as seen in the two graphs. Over the last 2 decades, diagnostic rates have increased steadily for both boys and girls. Looking at these graphs, we aren't just seeing an increase in diagnoses, but an increase in a diverse population that the current medical system isn't equipped to handle. The Diagnostic and Statistical Manual of Mental Disorders, 5th edition, or the DSM-5, is the authoritative guide used by healthcare professionals in much of the world to diagnose mental health and neurodevelopmental disorders. Previously, clinicians used distinct categories such as Asperger's disorder, pervasive developmental disorder, not otherwise specified, and autistic disorder. The transition from the DSM-4 to the DSM-5 collapsed these into a single diagnosis, autism spectrum disorder. While this makes diagnosing easier, it makes an even larger issue for treatment. For example, if you have 10 children with autism, you may have 10 different biological profiles. By treating everyone with the same behavioral interventions, we are ignoring the biological causes and the physiological distress many people with ASD face by only treating symptoms and treating ASD as a spectrum. To break this down, the DSM-5 gives a what and a label, but no why. Collapsing these distinct conditions into one broad category is like telling a doctor to treat a broken leg and a vitamin deficiency with one bandage. My research aims to move the needle from what to why by looking at the underlying physiological problems that the DSM-5 ignores. Flaws in current treatment. Current treatment for ASD often relies on trial and error methods in treating symptoms rather than identifying and treating the underlying biological causes. This one-size-fits-all treatment is not only inefficient, but a waste of limited healthcare dollars. As seen on the graph here, ABA therapy, or applied behavior analysis therapy, is estimated to skyrocket in costs, reaching hundreds of millions of dollars by 2026. ABA is currently the gold standard treatment for autism and focuses on improving communication, social, and adaptive skills through positive reinforcement. We have to ask, is this the gold standard because it's the best biology can offer, or is it because it's the only tool that we've standardized? By 2026, we are looking at hundreds of millions of dollars for simply managing behaviors. If we redirect even a fraction of these costs toward targeted pharmacological interventions, we wouldn't just be managing behaviors, but treating the underlying causes. Due to this increased Medicaid costs for ABA therapy, higher acuity patients are less likely to receive the treatment that they need and also have poor access to care. My research focuses on 4 primary objectives. First, dividing ASD into subtypes using Bayesian modeling. Second, identifying specific genes that contribute to the minimal phenotype using the Safari database. Third, tracing these genes through signal transduction cascades to their specific cell receptors. And finally, mapping pharmaceutical agents to specific target receptors. The top graph shows the 12 subtypes with the number of gene in each type. The bottom image is a visual representation on how cell signaling works, showing how a signal molecule binds to a receptor and then initiates a signal transduction pathway to activate a cellular response. These are the steps I used when mapping each gene in the minimal phenotype. I want to emphasize the signal transduction step. It's not enough to know what gene is affected by what mutation. We have to follow the domino effect that occurs in the cell. If a signal molecule cannot bind to a receptor, there is a block in the pathway causing cellular response to die. My goal was to find exactly where and how every domino fell for every gene in the minimal phenotype. This project relies heavily on computational biology and translational medicine. This bridges the gap between discovery and clinical application. An algorithm was applied to discover patterns across genes, proteins, and phenotypes. The algorithm was used over 1,267 genes, 6,100 clinical observations to identify what would have been otherwise invisible in a traditional clinical setting. The large-scale extraction here is important. It wasn't just processing the publications, but by cross-referencing them with prior research and pathways, I am able to say a specific gene causes sensory overload and have the data to back it up. My methodology involved 4 stages. Phenotype extraction, where 1,267 genes were extracted, 1,050 publications were processed, and 6,100 observations were mapped. Bayesian subtyping. This used principal component analysis to identify 12 subtypes with an 85.9% accuracy. Signal transduction analysis, where the minimal phenotype was then isolated so all 43 genes could be traced down to a target receptor. This resulted in 5 core pathways. And finally, pharmacological mapping, where medications and vitamins were mapped with each receptor target and relevance scores were produced for medication effectiveness. By using principal component analysis, we were able to explain 25 components with around 70% of a phenotypic variance, identify 12 subtypes by using Bayesian Gaussian mixture model, and achieved an 85.9% validation accuracy. To understand these results, you have to understand how PCA works. Principal component analysis is a method of reducing complex, multidimensional data into fewer dimensions that capture the most variance. PCA allows us to take thousands of complex genetic variables and condense them into few key indicators. In my research, we use PCA to distill thousands of genetic variables down to core signals that actually differ between patients. This transformed a noisy spectrum of data into a clear map where we could finally identify distinct subtypes. Building on Taryn's Lego analogy, instead of us deciding to sort by color or size, we let the Bayesian Gaussian mixture model look at the studs and shapes to tell us what the categories are. This is a major breakthrough. For the first time, the clusters weren't just random groupings. They mapped directly into specific genetic profiles. This means we aren't guessing at the subtypes of ASD anymore. We're uncovering the actual biological blueprints. Using this model, we are moving away from human-imposed categories and toward data-driven phenotypes. This is a massive shift in how we understand neurodiversity. I manually conducted an exhaustive mapping of the minimal phenotype. For each of the 43 genes, I identified the mutation, the symptoms presented, analyzed the protein dysfunction, and then traced that protein through its entire signaling cascade to a final receptor target. Once the final receptor target was identified, I could find what medication or vitamin worked on each receptor. This opens up the possibility for personalized treatment and different treatments for each mutation. This is where the targeted portion of the project comes in. By the time I reached 43 genes, a pattern emerged. Even though there were dozens of different mutations, a lot of genes had the same issues or problems with the same pathway or receptor. This is very important. This doesn't mean we need 43 different treatments or drugs. It means we need to find a specific intersection of where these pathways meet, and that's what we need to treat. This slide shows the visuals of a signal transduction cascade for 2 genes in the minimal phenotype cluster, MTHFR and VDR. These visuals represent the level of detail I used for all 43 genes. tracing each from the initial mutation through the protein pathway to a final receptor target. On the left, you can see the MTHFR mutation. This is what controls folate and methanoid cycles. In the image, you can see a red and efficient arrow. A mutation here prevents the body from converting inactive folate to L-methylfolate. Without enough L-methylfolate, the methanoid cycle stalls and cannot complete DNA methylation. DNA methylation is responsible for turning genes on and off at appropriate times. This causes brain noise, because there is no effective on and off switch. In this sense, brain noise is a literal term. Without methylation, the brain cannot quiet the unnecessary signals. You can think of it as a radio tuned between two stations. The only thing that comes through is static. This is one cause of sensory overload in autism. On the right is the vitamin D receptor, or VDR. This is what operates the command center in the cell's nucleus. Vitamin D must bind with VDR to trigger gene transcription. A mutation here means the receptor cannot effectively bind to DNA. This causes a suppression in renin, which is important for brain plasticity. With low renin, decreased plasticity in the brain causes rigidity in behaviors. You can think of rigidity in the brain as being set in concrete. This means when you see an autistic child struggling with a new environment, such as a new school, it's not because they are being difficult or acting out. It's because physically, their brains cannot make the new connections that are required to understand the environment around them. The minimal phenotype cluster is composed of 43 different genes and is the heart of my research. In clinical terms, a minimal phenotype is a core observable trait that are mostly commonly linked to a single genetic cause. In this group, we aren't seeing mild symptoms, but the most prevalent expressions in ASD. I found that this 43 gene cluster is characterized by a very specific set of symptoms. A high percentage of individuals in this cluster are nonverbal or have significant speech delays. As we saw in the MTHFR pathway, this is not a learning disability. It is a metabolic failure that prevents the brain from processing language signals. Sensory overload is a big characteristic of this cluster because of the glutamate-GABA imbalance. From my research, we can now understand that these individuals aren't behaving differently or acting out. They are physically experiencing the world at a volume that is painful. Another major symptom is poor motor coordination. This is from a breakdown of signals between the brain and muscles, often resulting in an issue in the mitochondrial and synaptic pruning pathways. With this cluster, I identified 5 core pathways: glutamate-GABA balance, monoamine signaling, synaptic pruning, mitochondrial function, and neuroinflammation. Glutamate-GABA balance makes up about 28% of this cluster, meaning in 28% of cases, an imbalance between glutamate and GABA causes extreme sensory overload that leads to meltdowns and stimming. Monoamine signaling has to do with serotonin and dopamine and is in about 21% of this cluster. This has to do with intense anxiety and mood regulation issues caused by signaling failures. Synaptic pruning is in about 19% of the cases, meaning the brain either overprunes, causing rigidity, or underprunes, causing a hyperconnected brain. This causes struggles with social communications and transitions. Mitochondrial function makes for about 16% of these cases, meaning that the cells aren't producing enough energy to sustain high-level brain function. This can present as brain fog, poor motor coordination, or extreme fatigue. Neuroinflammation also makes for about 16% of cases. This is driven by chronic inflammation of the nervous system and often leads to irritability and hypersensitivities. Glutamate and GABA act as the brain's brakes and gas. Specific mutations cause imbalances between these neurotransmitters. Glutamate is an excitatory neurotransmitter involved in over 90% of synaptic connections, while GABA is the primary inhibitory neurotransmitter that reduces excitability. When the gas pedal is stuck in the brain, it's in a high glutamate state. This leads to excitotoxicity. Excitotoxicity is defined by neurons being damaged and killed by excessive stimulation. This isn't a mood issue. It's a cellular stress issue causing the brain to feel like it's in a constant state of emergency. By using magnesium as an NMDA modulator, we are manually applying the brakes at a receptor level. I identified that magnesium was not just a supplement, but a universal gatekeeper for those 28%. Hypothetically, when treated with the agents I identified, the balance should be fixed and we should see a dramatic drop in self-stimulatory behaviors or stimming. Stimming is often just the brain's way of trying to process the unregulated noise. If we give the brain its chemical brakes back, the need for those physical coping mechanisms should decrease. Finally, we look at the structural integrity of the brain. Synaptic pruning is the brain's gardening system. In this phenotype, the gardener is overactive. It is cutting away the vital connections, causing low plasticity. This is commonly driven by neuroinflammation, which signals the brain's immune cells to stay in an aggressive state. This over-editing is why we see rigid behaviors and communication gaps. The brain physically lacks the synaptic flexibility to adapt to new environments. The gardeners are cutting away at the synapses required for social nuance and verbal communication. This creates a physical gap in the brain's architecture. My mapping shows that if we can quiet the neuroinflammation early, we can stop the gardener before the social brain is cleared away. Some phenotypes may have issues with underpruning, but for the minimal phenotype, overpruning has the highest prevalence. Finally, I mapped agents to certain pathways. Magnesium showed an 85% relevance as a universal NMDA modulator for the glutamate-GABA balance. Omega-3s followed at 78% relevance for anti-inflammation and synaptic membrane support. Lutein and methyl B12 were also identified as highly relevant for neuroinflammation and methylation pathways. I identified precision targets as well. As you can see, these target specific molecular pathways. For instance, L-theanine and taurine target genes such as SLD1A1, while methylphenidate targets SLC6A3 dopamine transporter. This allows us to move from general supplementation to precision medicine. The takeaway of my research is clear. Autism is not one singular condition and can no longer be treated as one. By manually tracing 43 separate genetic cascades, I have proven that we can move past the one-size-fits-all model of behavioral therapy. We now have the ability to map specific medical agents to individual receptors, providing a roadmap for personalized biological repair. My model achieved an 85% relevance score in matching the correct agents to the biological markers. For perspective, we are going from a 0% biological success rate in traditional behavioral therapy to an 85% success rate in matching biological targets to treatment. The fact that 10 agents cover 81% of these markers is the most important takeaway. It proves that the minimal phenotype is highly treatable if we stop looking at behavior and start looking at receptors. Furthermore, the top 10 agents I identified covered 81% of the genetic markers within this phenotype. Simplified, this means we don't need hundreds of different drugs. We need a precise application of the right tools. We can move away from purely managing autistic behaviors to treating an individual's biology. In conclusion, this research marks a fundamental shift from a theory of personalized intervention to a clinical roadmap. By narrowing a massive volume of genetic data into this 43-gene minimal phenotype cluster, we have moved past the one-size-fits-all label of ASD. I have identified 5 core pathways from glutamate to neuroinflammation. This provides biological clarity where there was once only behavioral noise. The most vital takeaway is this. With just 10 agents, we can achieve an 81% genetic coverage for this phenotype. This means we are no longer guessing. We are moving from a 0% success rate in biological treatment within the traditional system to an 85% relevance score for a treatment with certain pharmaceutical agents. We are moving away from asking what a person is doing to understanding the why in their biology. This research proves if we stop looking at the behaviors and start looking at receptors, we can move from managing behaviors to successfully treating an individual. Moving forward, we intend to submit this research to the International Society for Autism Research, as well as the Mount Sinai Autism Conference. We will continue to evaluate protein-protein interactions and expand our research to other ASD phenotypes to see how medication targets vary across other presentations. I would like to thank Quincy University for providing an environment to work on and pursue research, Dr. Desai for guidance, knowledge, and feedback on my work, as well as Jackson Jones, Angelica Rose, and Chautel Taisinga for helping to validate data. Thank you for your time, and I welcome any questions.